My name is Stephen Wang. I'm an associate professor at Yale. This work was co-led by my student Yubao Cheng, who's here with us today, and former students Mengwei Hu, Bing Yang, and Tyler Jensen. This work was done in collaboration with Professor Chongzhi Zhang's lab at the University of Virginia and was in part supported by the National Human Genome Research Institute, NHGRI. We know our genome is folded in 3 dimensions, or 3D, in a hierarchical manner across many length scales. These 3D genome structures control many essential genomic functions and also show intriguing dynamics and changes during development, aging, and many diseases. To learn more about the 3D genome, please refer to my talk last year at the Advanced Genome Technology Development Forum, now available on YouTube in the Genome TDCC channel. This short lecture covers a brief history of milestone 3D genome discoveries, and you'll see how technology advancements played a driving role in these fundamental discoveries. For example, I previously led the development of Chromatin tracing, a first-in-kind image-based 3D genomics technology to sequentially image, pinpoint, and distinguish numerous genomic regions along individual chromosomes in single cells, essentially super-resolving the chromatin folding conformation. This technique is based on a highly multiplexed version of DNA fluorescence in situ hybridization, or multiplexed FISH, and it enabled direct tracing of the 3D folding organization of DNA in single cells in situ. Despite the powerful techniques and all the beautiful discoveries in the field regarding how the genome is organized and folded in 3D, and how 3D genome is associated with aging and diseases, a key question remains. That is, what controls the 3D genome? There are potentially many regulators of the 3D genome. Our vision here is if we can identify many 3D genome regulators, they may serve as new therapeutic targets to halt or even revert harmful 3D genome changes in, for example, cancers and aging. But a key limitation here is there is a lack of method to allow efficient discovery of regulators of the 3D genome, especially across the many different length scales that I talked about. In this work, we set out to address exactly this technical limitation.
To tackle this technical limitation, we developed the perturb-tracing that enable high-content screening of multi-scale 3D genome regulators. This technique combines 3 cutting-edge technology modalities. The first one is pooled CRISPR screen technology. This technique uses lentivirus to introduce individual gene knockout or perturbations into the single cells with a pooled manner, enabling the parallel perturbation of many genes across individual cells. Next, using chromatin tracing, we can visualize how 3D genome conformation changes or remain unchanged across this pool of cells in response to different perturbations. Then, very importantly, we need to figure out in our image which cell carry which perturbation in order to link the genetic perturbation to 3D genome phenotypes. For this purpose, we paired each guide RNA a barcode RNA that can uniquely map the guide RNA identity. And we developed a cellular barcoding and in situ decoding technique called BARC-FISH, which can read out this barcode digit by digit. This allows us to determine which cell contains which barcode and its associated perturbation. This then led us to link the genetic perturbation in each cell to the changes in 3D genome conformation revealed by chromatin tracing. In our CRISPR screen, we include 420 guide RNA, including 10 non-targeting control guide RNA and 410 guide RNA targeting 137 selected genes, most of which encode nuclear proteins. Here shows some raw data of our CRISPR screen. In this field of view, in this single cell, we can read out the barcode digit by digit. It's a 10-digit ternary barcode. Each digit's ternary value is represented by 3 colors. Based on this information and the design of our codebook, we can know that this cell carries a barcode corresponding to a guide RNA targeting a particular gene. Using chromatin tracing in the same cell, we can then analyze how this perturbation influences 3D chromatin organization. As a comparison, in a conventional perturbation experiment, people usually perturb one gene and image one target to observe phenotypic changes. In our work, we have 420 perturbations introduced into single cells in a pooled fashion and have 30 imaging targets imaged in each single cell. This is equivalent to 12,600 of conventional experiment. This high content nature of our screen allows us to effectively discover novel regulator of 3D genome architectures at different length scale. For example, at the scale of topologically associated domains, we identified a list of novel regulators that can lead to increase or decrease of adjacent tag distance. Here you can see CTCF and NIP-BL, which are known 3D genome regulators involved in the famous loop extrusion mechanism, serve as our positive control, and they show the expected changes. At the scale of AB compartment, we identified the novel regulator of intra and inter-compartment contacts. At the scale of the chromosome territory, we identified a novel regulator that leads to global compaction or decompaction of the whole chromosome territory. And then at the scale of the whole nucleus, we identified the regulator of the nuclear texture feature, which is represented by the dipistinian homogeneity of the cell nucleus, and also the nuclear shape, whether it is more spherical or multi-lobed. Taken together, our screen identified 21 novel regulators of 3D genome organization across multiple length scales. Our dataset provides a valuable resource for further exploration of 3D genome regulation and has been uploaded to the 4DN data portal. The computational analysis code generated for this study is also available on our lab website. Among our top hits, one of them that caught our eye is CHD7. CHD7 or chromin domain helicase DNA binding protein 7. CHD7 loss of function mutation causes charge syndrome, which is a rare but severe congenital disorder. The known function of CHD7 is a chromatin remodeler that can promote local chromatin accessibility through chromatin remodeling. Our work unexpectedly identified that at a long range, CHD7 is a chromatin compactor. We validated this result by both knockdown and overexpression. We further validated this result in different cell background and genomic context. So CHD7 has contrasting 3D genome organization effect at short and long ranges. We similarly validate two other top hits with individual perturbations. The rich information and high content nature of our screen readily enable the discovery of potentially functional linkage between different 3D genome features. Among all the top hits we discovered, we found a significant correlation between their effect on chromatin compaction or contact and the nuclear sphericity. It seems chromatin compactor in general helps more spherical nuclear shape. And indeed, when we individually knock down CHD7, the nuclear sphericity level goes down and the nucleus became more multi-lobed.
It's important to note that CHD7 belongs to the family of chromatin remodelers with similar domain layout, including CHD8 and CHD2. These are genes associated with autism spectrum disorder. Overall, most of our top hits are associated with human diseases. So we think our results really open up many new avenues to study 3D genome regulation in diseases. As a summary, we developed an image-based high-content genetic screening platform termed Perturb-tracing that uniquely integrates real 3D genomics by chromatin tracing, pooled CRISPR screen, and a novel cellular barcoding and in situ decoding technique termed BARC-FISH. The high-content nature of our screen enables efficient discovery of regulators of 3D genome architectures across a wide range of length scales. As a first demonstration of our new technique, in this work, we only screened a small portion of the potential 3D genome regulators. But in the next step, using perturb-tracing, we will be able to build the complete 3D genomic regulatome in different health and disease contexts. With the newly discovered regulators, we can also start to test whether we can use them to manipulate 3D genome as a new class of therapeutics to halt or revert deleterious 3D genome changes in diseases and aging. To access the perturb-tracing technology, a key requirement is to have an automated imaging and fluidics platform capable of chromatin tracing. We welcome researchers who are interested in setting this up to reach out to us, and we will provide instructions on several potential solutions of this setup. Thank you for listening.